All right. Hello, everyone. My name is Vivian Zhang, and I'm the Business Development Associate at Omicsoft. And today, we're glad to have Matt Newman to present some of the exciting updates in land, both including OncoLand and DiseaseLand. So for today's webinar, it will be about 40 to 45 minutes. First, we will turn to the presenter for about half an hour presentation, and then afterwards, we will have a Q&A session. So please post your questions may you have at any time during the presentation. And during the Q&A session, our presenter will pick up the questions. If your question is not addressed during the webinar, or if you have any follow-up questions, we will definitely get back to you, or you could email Omicsoft at any time. Your question will be addressed. All right, so now let's get started with the presentation, and I will turn to the presenter. Welcome, Matt.
Okay, thanks, Vivian. Most of you, I'm looking at the attendees. I've seen most of you before online, so you know who I am. This is Matt Newman. I'm VP of Business Development. My goal today is just to give you a high-level overview of some of the new features in OncoLand and in DiseaseLand. And so I'll try to go relatively slow, but I probably won't cover everything, and we'll take maybe 25 to 30 minutes. And as most of you that have been on these calls with me before know, if you put a question into the box, I'll look at it as I'm talking and switching back between the slides and the software itself. So just to get started, just high-level overview of both OncoLand and DiseaseLand. This I just took straight out of the emails, but I thought I would cover some of these in more detail. Starting with OncoLand. It's the official release of the B38 lands, including TCGA, Cancer Cell Line, Encyclopedia, GTEx, Blueprint, and then the Sanger Land if you have access to that. So let's take a look at that. As you guys, most of you know, all of our lands were based on the B37 genome in the past. And then last year we released the B38 genome, or earlier in the year, I guess, the B38 genome for these same lands, but only for the RNA-Seq data. So I think there was only a subset of the users that were actually using it. So what you'll see if you type in, for instance, TCGA into your land search box here, is you'll see it now a section for OncoLand B38 as well as OncoLand. And so now I have the B37 version and the B38. We felt that B38 was mature enough from a genome perspective and from an annotation perspective to work now. So just click my B38 genome, and then I do my search as usual. Yeah, as usual, the clinical data and everything I think matches between the two lands, and the same thing applies to the Cancer Cell Line Encyclopedia and the other lands that we've listed. So just for overall sake, let's do a search, and we'll see that we now have the usual screens for the land. We can provide more details. I don't know if there's going to be questions about it, but I can provide more details offline as to what we've done for some of the other data types. But you will see all the different data types that you're used to seeing, except everything now is on the newer genome. So there's a possibility that some of your GNF PKM values are slightly different, or potentially mutations are different, depending on any changes in the genome. But I think it adds some added value, especially for TCGA. So along with that, one of the things that I wanted to talk about was TCGA in general. As most of you guys that are in oncology probably know, it's starting to wind down in terms of new data. And so one thing that you don't see in our OncoLand updates is any new TCGA data, with the exception of updated clinical data, which we'll of course continue to do, and some updated copy number variation call data in the TCGA lands. So one thing that we've started to focus on is how to add value to TCGA itself. I think there's plenty of value in OncoLand with all the other stuff that we're adding, but for TCGA itself, one thing that we did this time, that for those of you that have seen my personal demos, I actually did maybe a year or so ago, is for each of the tumor types where we had normal and tumor, we actually created comparison data. So for those of you that have used DiseaseLand or have used the OncoGeo Land, you're familiar with comparison data. But what this is essentially a set of, for RNA-Seq, it's going to be DE-Seq2. It's a set of comparisons comparing the tumor and normal samples. So what you'll see in the land itself, there we go, is when I search for a gene, whether it's in B38 or B37, we'll do it in B38 since it's already open here. You will see a comparison tab now right in the middle here. So let's close everything else so you can see what I'm talking about. And in this case, all we have right now is disease versus normal. I can imagine in the future we're going to have other types of comparisons as well. You know, you can compare mutant versus wild type. There are different subsets that you can do. But you know, essentially, what this shows me, if you're not familiar with this view, is for each of the 24 tumor types. So each dot is a comparison comparing tumor versus normal. The x-axis is the fold change, the log2 fold change, and then the y-axis right now is organized by disease category, which is kind of like a high-level disease category. So you'll see, you know, some of the categories have multiple tumors in them. And then the size of the dot is indicative of the p-value. So the larger the dot, the more significant the p-value. So what this tells me off the bat is that when comparing all of the tumor versus normal in whatever this dot is, it's highly overexpressed. So we could of course mouse over it to find out, or if you select it, give it a second, and it should link to the underlying data. So what it'll show you is what was actually compared. So this was comparing tumor versus normal for glioblastoma down here in the brain, of course. There's no treatment for this for any of the TCGA data, and then it's going to show you a nice plot of the comparison here, which you can double click to go full screen, and of course export as well. You know, the same thing will work if I select any of these other ones. You know, if I want to know, you know, what this one is, and then it's going to give me that, and you can see as I switch, it'll show different ones. If I pick more than one, it'll give me a list here that I can scroll between and see the different plots, which is kind of nice. This is the what we call sample level detail. Then we also have the underlying details for whatever's selected at the gene level. So it's showing me, you know, what was compared, what the fold change is, what the p-value is, and so forth. In some cases, there looks like there is no p-value. I'm not sure why. The other thing that you can do is after selecting one or more, so let's just pick one. If we come down to the bottom left and say Browse Selected Comparison, that's going to take me to the full set of the DE-Seq data comparing the tumor versus normal in GBM. So we'll give that a second to load. Again, for most of you that have seen either DiseaseLand or you've used the OncoGeo data set, you're pretty familiar with this functionality, but for some of you guys it might be new. So let's let that run. And so what it gives me is a volcano plot showing me all the different tumor versus normal. So I can quickly see that in GBM, that whatever this gene is the most differentially expressed, highest p-value, and it's set up so it'll show you. Well, it looks like the gene IDs. It should be the gene symbols, but you can get that information down here, and you can see the list of genes. And of course, all the other functionality that is normally available for any kind of comparison will be available here. So if you just want a giant table of all the comparisons, you could do that. Different summary statistics. There's one that shows you significant genes. So if you've selected more than one comparison, it'll give you a nice little plot of ups and downs if you're trying to look for similarities between the different tumor types. Again, I think this is one area that we can explore more in the future. I think there's different mutations that we can compare. So it doesn't have to just be this this category of disease versus normal, I think we can do all kinds of interesting things here as well. So this is the first step for doing any of that. And I think I know from talking to some of our users that have used the version I created a couple of years ago that it is useful. There's the downside of it being pretty generic, where you're taking all of a tumor versus all of a normal. And I understand there's not all of GBM samples are exactly the same, but it at least gets you a high-level overview of what's changing. All right. So going back to OncoLand, additional samples in Target and Blueprint. I just want to point these out again in case people are not aware of them. So let's just go to Target. The Target data set is a whole set of blood-borne cancers, childhood cancers, and so I'm not sure that just from looking at individual companies' logs whether or not how often the Target data set is being used. But if you go into if you Google Target, you'll see all the different tumor types. But you can see it's a whole lot of different blood-borne tumors: ALL, AML. I think it's Wilms tumor, and it has any data. It has RNA-Seq. It has some mutation data, some copy number data. There's a good amount of RNA-Seq though in the blood-borne samples. There's about 1,300 different RNA-Seq samples in the different tumor types here. So this is a pretty valuable resource in addition to TCGA, and they continue to update this resource as well. I didn't put it in my notes, but one update that we did over the last month or two that applies regardless as to whether or not you've done the updates. There was a blog about it that Vivian sent out, I think, last week or the week before, but it relates to filtering. So this is I think a really nice improvement in the system, in that when you're exploring this main land tab, if you were, let's say, interested in just a particular tumor type, so let's say lung, there's a lot of times where you're exploring these filters and you're figuring out how many samples are available, and then you go to do the search. In the past, any filter that you had on was entirely lost after the search. That's changed over the last couple of weeks, and so now that filter is actually propagated down. So you'll see that it automatically filtered for lung. For the long-time users, this might be a little bit of confusing change, but I think in general it makes the workflow a little nicer, in that you can use your land of interest. You can query for whatever subset, and then when you search it, it already automatically filters for that. You can of course always reset it, but I think it's a pretty good update, in that it'll save you some of the extra time, especially in the past our recommendation was to create a sample set and then filter for the sample set, and you can of course still do that if it's going to be a long-term need, but it's I find myself looking to see if there's particular clinical covariates, if there's any samples with a particular clinical covariate, and then filtering for it, and then I get to the end and I have to do it all again once I do my search, and this will just save you a little bit of time there. Okay. All right, so that was Target. Blueprint, just covering again, is out of Europe. We've now released both a B37 and a B38 version of Blueprint. Let me just close some of my windows here. And so there's some additional data in here. This is a good, if you're not familiar with it, this applies probably to both OncoLand and DiseaseLand, but it's a good data set. We've put in all of the RNA-Seq data, so there's about 250 total RNA-Seq samples across all these different cell types. Most of them are normal samples. I think there's a couple oncology samples in there. But if you're looking for expression of a particular cell type, this is a pretty good place to start. You can see when I type in my gene and do a search, it just returns all the different cell types. And this has been updated with some additional samples that weren't there previously. Close that. Close target. Okay. And we don't want to skip ahead to links. All right. So we've also added about 5,700 new samples into our TumorMutation land. So a lot of times I'll get requests from customers that are asking me whether we can include data that's in one paper or another. That's going to be, usually we put that data, if it's not RNA-Seq data, into this tumor, all-encompassing TumorMutation land. And so you can see that they're named via the typical naming technique that we've used, like PRAD would be prostate adenocarcinoma, and then there's a dash, and then it'll tell you who wrote it or where it came from. If you go into the sample details window, you can get a little more detail about where it came from, and then also what the disease is and everything. I tend to use the disease column here rather than the tumor type column, just because it gives a little more detail. So this is saying triple negative breast cancer, for instance. What this land consists of are almost entirely DNA-Seq data, somatic mutations. There are about 314 copy number samples, and then a few expression samples, so not RNA-Seq, but just standard expression. And it, of course, works the same way as all of our other lands, in that you query for a gene, except that it's really focused on the DNA-Seq side of things. So you get this view that basically shows you the percentage of mutated samples, and you can see lung adenocarcinoma from a few different projects is clearly sticking out here. There's a view under DNA-Seq called Genome Browser Somatic Mutation that gives you kind of a landscape view of where the mutations are, and you can see this dot here is clearly, there's 19 samples, and it's L917R change that's found in a number of these different, a few different lung adenocarcinoma projects. And there's, of course, other ways to explore this data, but this is just the high-level view of it. Okay. New samples and comparisons in the OncoGeo Land. If you're not familiar with the OncoGeo Land, what we've attempted to do is basically have the same type of lands that in oncology that we have with DiseaseLand, where we're taking data sets from Geo and the sequence read archive. We're with a heavy focus. If you look here, it's 2,200 RNA-Seq samples and about 5,000 expression samples, mostly from Affymetrix. But what we've done is we've gone through the individual Geo projects. I can go into the select view and go to the projects to see, and you can see there's about 249 total samples here. If I just view filtered table, you'll see the list of all these projects. And so what we've done, it's similar to what we've done with DiseaseLand, is for each one of these individual projects, we've reprocessed the data. RNA-Seq is going to use our standard pipeline. The expression analysis is also using a standard pipeline. And then we're coming up with what we call comparisons. So similar to what I showed earlier for TCGA. But in breast cancer, there's a couple hundred comparisons comparing different things. So in this case, it's treatment. The treatment comparison, treatment transfection, treatment versus control. Whereas in other categories, it might be, if we look at the legend, you can see there's a whole set of disease versus normal, responder versus non-responder, and so forth. And so again, if you do your query in this land, you're going to get back that view that I showed earlier with TCGA, except that it's going to be with all the data sets that we did from Geo and the sequence read archive. And so, we're not actually seeing a high overexpression in any kind of nervous system cancer, but it could be reflecting what's actually out there. But we are seeing, oops, I clicked something random. We are seeing, a decrease in breast cancer, for instance. So all of these dots here, I think, are breast cancer, and ER positive versus normal, just normal tumor versus normal, triple negative versus normal, and then HER2 positive versus normal, all showing the same profile. You also have the ability to get to the underlying data. So we have the data at the project level, the overall gene FPCAM level, if you want to try to compare everything, and then a number of different views that break down, disease versus normal, treated versus control. All comparisons will show you a plot of everything that's going on in that. So you've got your diseases here, followed by your treatments, and so you would likely use some sort of filter here to figure it out, to kind of narrow it down. So like I might say, just use tissue for breast, and so we narrow it down into the different types of breast cancer, and you kind of get an overview of what's going on with the different treatments versus some of the different diseases. Okay. Go ahead and close that. All right. There's 1,750 new samples in the ClinicalOutcome Land, and again, not sure how, I've talked about it before in some of these webinars, but I'm not sure how often they're being used. But there's a ton of data out there that contains survival information, and so what we've done is tried to collect all that expression data. There was a paper that was written that did something similar. We're not completely using the same data. Some of that was not accessible. But the main goal was to grab as much expression-related data, so there's about 11,000 samples, and then and put that in. So every one of these samples has some sort of survival information, and so when you search for a gene in this land, it's actually going to return a slightly different plot. It's going to return a survival plot. I always like to go into the task tab and show my summary information. So what we're doing is basically grouping these into upregulated, downregulated, and no change. And this is across all 11,000 samples. But for instance, if you went into nervous system cancer, you'll see a clear change in the p-value. So upregulation of EGFR is leading to death, as opposed to the downregulation or no change. So again, just another area that we're consistently adding to, that I wanted to make sure everybody was aware of, and there's a whole lot of new data in that land since the last update. All right. And then I'm not going to show it, but I touched on the clinical data and the CNVCall data being updated in TCGA. So with that said, I think I'll move from there into DiseaseLand, and I'm not going to shortchange DiseaseLand, but I think I'll do a couple different examples. But I mean the main, and actually I left one out here that we'll talk about. The main thing to keep in mind that we're doing with DiseaseLand is continuing to add a ton of RNA-Seq and standard samples into each of the different lands. So as you guys know, we have Human DiseaseLand, Mouse DiseaseLand. For those of you that are still on just ImmunoLand, there is the possibility of upgrading you guys to the full DiseaseLand, but you don't have to. But basically what we did is we merged all of the ImmunoLand data with the cardiovascular and metabolic data, with the idea that we now have two major disease-related lands. We have oncology, and then we have essentially everything else. The other thing that we've continued to do is add additional samples into our single-cell human land, as well as our, I should say, single-cell mouse land. So you can see there's about 3,916 new single-cell samples in the human and over 5,000 in the mouse. And then finally, we have about now, DiseaseLand itself now contains over 67,000 human samples with 3,200 different comparisons from 1,000 plus projects. So we're continuing to just add to each of these lands. So take just a quick look at some of them. So if we go into our DiseaseLand collection now, for whatever reason, my mouse one is doesn't look right this morning. So normally you have a mouse disease B38 and a human disease B37. We're just going to take a look at human here, and you can see all the different types of samples that we're now hitting across different disease categories. Again, you can always change this to something like disease state to try to go in and find more detail. There's a whole lot of normal samples, as what this big section is. But one question I get a lot is, how do I find whether or not there's any samples in my disease of interest in there? And what I recommend using is the disease state category, and then the sample source category is interesting for cell types, if you're looking for a particular cell type. And there are a few other ones that sample pathology can sometimes be interesting as well. One of the things I find myself doing is just emailing our support team that then talks to our curation team. So if you're wondering whether or not there's a particular sample type, shoot us an email. We can probably lead you to where to go to. But if you focus on those categories, you'll for the most part do pretty well. I can show again the searching. I won't use EGFR for DiseaseLand. Just do Serpent B7. And then, again, the highlight here is that we continue to just add data, including RNA-Seq data and expression data into this land. So I think it's becoming pretty large and has a ton of data in here. And then what I'll talk about, so again, this is the same plot we've looked at a few times, showing the different dots being individual comparisons and getting an idea of where things are up and down regulated. So I'm seeing it upregulated in arthritis, for instance. And then when I select it, it shows me the details of the plot, of the project itself. And you can see that it's juvenile idiopathic arthritis versus normal in the inactive GQ1. As opposed to some of the ones down here. I'm guessing that there are cancers that have other diseases as well, and that's why we're seeing cancer in DiseaseLand, but I'm not actually sure. And again, of course, I can look at the treatment data and responder versus non-responder and all the different comparisons that we've looked at in the past, and most of you guys are aware is in the system. I'm not sure how many people attended some of the gene set analysis webinar that I gave a month or two ago, but one of the things we really thought about over the last couple of months is how to add more value to all of the information that's in the land. So you can see even today I've gone through 8 different lands here, maybe more, and so you're trying to figure out which land to go to and how to query it. And one of the common questions people had is, I have a gene signature, which disease is it hitting? Which cell type is it hitting? Which treatment is it hitting? And so we added a gene set analysis functionality into Array Studio a few months ago. It requires you to have a server project, so you just create a new server project, and then it's in this integration menu. It says gene set analysis. And what it does is it pulls all of the data from the land. So we've classified things into disease land, into mouse disease land, molecular signature database, OncoLand, a whole set of reference data from the blueprint and the GTEx data sets and so forth. All the single cell line data is in there as well. There's one land that I haven't talked about yet, the links land. That'll be in here by the time the release happens. And basically, you give it a list of identifiers. So you either have a list of genes that are, that have no direction. You have a list of upregulated and downregulated genes, or you even have a list of genes with p-values and some sort of rank, like a fold change or copy number value or whatever. And you walk through this module, and you would paste in or type in your genes depending on which option you were working with. And what you end up getting back, I'll just skip to the end, is a couple of different tables. One is this gene analysis result table that has a huge, basically a giant table that I've got about 20,000 rows here. Each row is showing me a p-value and what the overlap was with my input data set and with this other data set. So I had my input was the down direction, and it's with GSE 11292, and we're seeing a p-value of 0.009. Although the Bonferroni p-value it makes it pretty much it makes it 1. This table is good, but what we added over the last couple of months were a couple other additional tables, and one of them is this enrichment score versus p-value. So what we did is we summarized this table to give you an overall idea of which gene sets are being hit. And so I put in it wasn't random, but I put in a kidney-related gene set into the system this morning, and what it pops back is a volcano plot that's very similar to if you're doing an expression study. And so on the x-axis is the score, and a positive score indicates correlation, and a negative score indicates anti-correlation. So if I want to see what my gene set was correlated with, I can select all of these up here, or one of them. And so what it's going to do is it's actually going to show me a volcano plot that'll show me how many genes were in my query list. 318, 182 of them were overlapping, and then there was 318 were not overlapped from the query, and 20 were not overlapped from the target, but there was an overlap of 182. We can see it came from the OncoGeo project, and we can see that it's renal clear cell carcinoma, primary tumor versus solid tissue normal. So what it's telling me is that I'm in a positive direction, a correlated direction. I'm hitting an overlap with this gene set. And this is working across all the different lands that you subscribe to. So not just, if I have OncoLand and DiseaseLand, it'll search over all of those. It'll also go in the opposite direction. I'm sorry, it'll also do some of the body map lands as well. So you'll get cell type specific expression, you'll get tissue specific expression. So this extremely negative one is actually telling me that it's anti-correlated with the kidney cortex from GTEx. And so if we look at a couple others of these here that it's anti-correlated with, we're seeing some other, let's see, we're going to scroll over until we see the name. Here we go. Some other kidney-related stuff, infliximab, which I'm assuming is a treatment that apparently sends it in an opposite direction of kidney cancer. So anyway, there's a whole lot that you can do to explore here. Finally, we have another plot that was added over the last couple of months that basically breaks it down into how your gene set interacted. So how many overlap genes in the up direction, the down direction, and the overall direction. And so if you want to dig into more details about the individual individual sets of directions, you could do that as well. I like this plot combined with the volcano plot and then you go back to that full table as the final link. But this to me is probably one of the biggest adds over the last 3 months in terms of value, because there's just so much data in DiseaseLand and in OncoLand that it's sometimes overwhelming. And this is one way to kind of dig down into it. And so once you find your data sets of interest, then it makes it a little easier to then link back to the lands and explore those data sets in more detail. All right, it doesn't look like we have, let's see, do I have questions? No questions, okay. The last thing I want to talk about is a land that I didn't even include in the description, but it's the links data set. So I'm not sure how many people are familiar with this data set, but let's just Google it here. Links Geo. So this is a data set that contains, it's the Library of Integrated Cellular Signatures. It's an NIH program where they basically took different types of treatments and other different types of profiles across a number of different cell types. And so we have expression data on all of it. There are a ton of samples in this data set. And it's not something that I've personally spent a whole lot of time with because we just got our hands on it over the last couple weeks. But if we look at the land itself, you can see we're organized here by treatment group, and you're seeing the different types of treatments that were done here. If we look at actual comparisons, there's 28,000 total comparisons across all these different tissue types that were actually done in this study. And so you get a full change in a p-value for all of them. This will be included in the gene set analysis as well, so you'll be able to take your list and then return the top hits from this, which I think will be pretty interesting. For the short term, and that should be done before you guys get your hands on it. I just don't have it yet. But for the short term, just to show you what it looks like, because I think that there's a lot going on. If you query for a gene, for instance, this is what it looks like. You have thousands and thousands and thousands of comparisons, and selecting things will show you exactly what the profile looks like. So this is comparing TGX 221 versus DMSO in human 83375 cells, which is a skin cancer, as a melanoma cell. I can, of course, I narrow this down for anything from a cancer type to a normal control, and we can see all the different comparisons that were made. A ton of data here. I think a lot of the value of this is going to come from the gene set analysis, and then also just in general being able to get your hands on all this data. But using the filters here is going to be extremely important, and this is included as part of DiseaseLand in the upcoming release. All right, so I think I spent my 35 minutes. I'm not seeing any questions so far, but maybe we'll give it a minute, and then I can answer, and then we'll call it a day. Okay. Well, if there were additional questions, most of you guys have my email address. You should have Vivian's, because she sent out the webinar information. Feel free to shoot us an email with any questions, and then, as always, the support team will contact you in the near future with how to update. There was a question about whether the Achilles data was in there. I don't think the Achilles data is in the land, and I don't remember the reason why. I think we looked at it, but the person who asked me the question, I'll follow up with you later. Any other questions? Oh, there's one. If I have a gene and would like to know the correlation of it to a treatment in length, I think that you're going to basically do something similar to what I've done now, which will basically give you an idea of whether or not the treatment of that gene is increased. But I guess it depends if you're looking for a specific treatment or what, but all the treatment information is in here as well. So you can filter for, a particular treatment and then get back the expression and what it looked like comparing the two. Give it another 30 seconds and then see if we have, it looks like people are starting to drop off. Okay, well, thank you everybody for attending, and as I said, just shoot me an email if you have any questions.
All right.
Vivian,
Thanks, Matt, for the Thanks, Matt, for the presentation. And we will follow up with the recordings shortly after the webinar. And I think in the next couple of weeks, we will be delivering the land updates to each of our clients. So stay tuned for the updates and try it out with the new data samples and new features. And we look forward to seeing you next time during the next webinar. So all right, so that concludes today's webinar. Thanks again for your attendance, and hope you enjoy the webinar and have a nice day.
All right, bye everybody.
Bye everyone.